Hello and welcome, my name is Ross McKenzie. I am an undergraduate student from the University of Bath, currently on placement at the University of Arizona College of Medicine, working in the laboratory of Dr. Herbs Kralowitz. I would like to thank ITSOG for awarding me a scholarship and allowing me to present. Mobiluncus mulieris and Eggerthella are two of the relatively understudied bacteria associated with the polymicrobial condition bacterial vaginosis. BV has been linked to a range of adverse obstetric and gynecological outcomes, including increased risk of STI acquisition, preterm birth, and pelvic inflammatory disease. Our objective was to assess the individual contributions of both of these bacteria to the cervical microenvironment through immunometabolic analysis. To do this, we utilized our immortalized human cervical epithelial cell line in conjunction with microcarrier beads and our rotating wall vessel bioreactor to generate 3D cervical cell aggregates. These aggregates were then infected with bacteria and imaged with scanning electron microscopy, whilst the cell culture supernatants were analysed with immunoproteomics and global untargeted metabolics analysis. Our scanning electron microscopy imaging demonstrated we can effectively colonize our 3D cervical cell models with both of our bacteria, as well as indicating that our bacteria interact with the epithelial cell membranes as well as other bacteria. Our immunoproteomics analysis revealed that Mobiluncus mulieris significantly elevated IL-6, IL-8, and TNF-alpha, which are all linked to preterm birth, as well as MCP1, whilst Eggerthella significantly elevated the production of IL-1-alpha. Our global metabolics analysis revealed 340 known metabolites for each bacteria, and using principal component analysis, these were condensed into metabolic profiles for each bacterial biological replicate. These bacterial biological replicates clustered distinctly and showed clear separation between infection conditions. The significantly altered metabolites by both bacterial infections were predominantly associated with the amino acid and lipid superpathways. Delving into the lipid superpathway, we saw Eggerthella significantly altered twice the number of lipids as Mobiluncus mulieris. These lipids were primarily sphingolipids and glycerolipids, which have been associated with membrane function and signaling. On the other hand, Mobiluncus primarily significantly altered long-chain fatty acids, which have also been associated with membrane function, but also energy metabolism. We also investigated BV-related metabolites and found Eggerthella significantly elevated multiple biogenic amines, including putrescine and cadaverine, as well as their precursors citrulline and ornithine. On the other hand, Mobiluncus significantly altered the levels of succinate and nicotinamide, which are related to energy metabolism, as well as 2-hydroxyglutarate, cysteinolglycine, and cysteinolglycine disulfide, which are all associated with oxidative stress. We also found that both bacteria significantly altered the levels of sialic acid, indicating they both have sialidase activity, which could play a role in how they colonize the female reproductive tract during bacterial vaginosis. In conclusion, Eggerthella significantly elevated multiple biogenic amines relating to elevated vaginal pH, vaginal odor, and vaginal discharge, which are all part of the Amstel criteria, which is used to clinically diagnose women with BV. Eggerthella also significantly altered the levels of lipids relating to epithelial barrier function. In contrast, Mobiluncus mulieris significantly elevated pro-inflammatory markers relating to preterm birth, as well as significantly altering metabolites relating to energy metabolism and oxidative stress. Future studies will need to be done to elucidate the roles of these two bacteria in polymicrobial infections like bacterial vaginosis. Thank you all for listening.